Alright, this is DNA synthesis and repair. So what are genes made of? The key to the study of chromosome theory was DNA and protein. So scientists basically wanted to find the code of life. And what they had was protein, which composes of 20 different amino acids, and DNA, which composes of 4 types of nucleotides that are different in minor ways. So in the 1940s and 50s, experiments were done to identify DNA as an inheritable material. And one of the things that were used was bacteriophage, and they did this to examine DNA and protein. What they wanted to find was what was virus transforming when it was, or what was virus injecting into a cell that was creating new viruses to form. And one of the first experiments we had was with Oswald Avery's experiment. And basically what he did was he took a bacterial cell and he heated it up to break it open and kill it, pretty much, to expose the living cells and alter its traits. And what he found was that when molecules were released from this dead bacteria and transforming live bacteria. So he wanted to know, what was this transformation? What he did was he took a bunch of test tubes and he put the bacteria into these different test tubes and wanted to test what would happen if he removed certain things. So he removed DNA, protein, and RNA, or DNase, RNase, and protease. And what he discovered was really cool. What he found out was, okay, with protein and RNA, the bacteria transformed, but when DNA was removed, it was completely dead. There was no transformation. And that concluded that DNA is basically transformation or info-bearing molecule. Which led to Hershey-Chase experiment. Martha Hershey and Alfred Chase in 1952 decided to experiment with this as well, and they took a T2 bacteriophage and they wanted to examine the role of DNA. And this is kind of infamously known as virus, or putting a virus in a blender. And so basically at this time, they didn't really know too much about what was going on in a virus. So they said, okay, we're going to take DNA and protein and we're going to see what happens when we put a virus in a blender. I mean, interesting stuff. Anyways, they labeled viruses into 2 groups, P32, which was directly associated with DNA, and S35, which was associated with protein. And they decided to infect E. coli and put it in a blender to centrifuge it. And basically forcing the cells into pellets, they found out that the DNA created a pellet, which meant that viral genes consisted of DNA over protein. So these early hypotheses about DNA synthesis led to other types of hypotheses. So we have different types of hypotheses that were created. The first one, and most known one today, is called the semi-conservative replication. When the double helix splits, an old strand is copied to generate 2 new chromosomes. So in short, new chromosomes will have 1 old and 1 new. And this is the one that is used today. Other hypotheses that were acknowledged included the conservative replication. An original chromosome is copied but remains unchanged. So basically, a new chromosome is basically new. And then you have dispersive replication. This process involves generating 2 new chromosomes with new and old sections of DNA mixed together randomly. So basically, new and old sections of DNA mixed together randomly. So what is DNA replication? So this little purple circle you see is called a DNA helix. And what's going to happen is the DNA helix is going to go to an origin site. Origin sites vary between eukaryotes and prokaryotes. Eukaryotes have a lot of origin site bubbles, and prokaryotes only have 1. In this example, we see 1. A helix job is basically to separate the DNA or break its hydrogen bonds. And while this is happening, SSBP proteins, or single-stranded binding proteins, will latch onto DNA strand to stabilize it. In order for new DNA to replicate, you will need something called a primer. The primase or primer is going to latch onto a strand and create temporary RNA nucleotides. This will later be replaced by DNA by polymerase 1. There's also something called a topoisomer, which is not shown here, and basically while all this is happening, DNA is twisting a lot, and to stabilize it, the topoisomer is added to this process. Its job is to keep the heavy twisting organized and control. There are two strands, so two strands are basically being replicated. We have the leading and the lagging strand. In this example, we can see that first that we have the primer of RNA, and then you see DNA polymerase 3, which is going to help pretty much extend everything. Overall, we're going to focus on the lagging strand. However, it's important to note that the leading strand is always going to move towards the fork, and the lagging strand is always going to move away from the fork. Overall, the lagging strand is going to compose of DNA fragments called Okazaki. And DNA polymerase 1 and DNA ligase is going to eventually connect everything as well. And that's what we see here. So again, leading strand toward replication fork, lagging strand away from replication fork. And then DNA polymers kind of extending everything. DNA polymers coming in to exchange the RNA for DNA. And the ligase basically connects everything. And what's interesting enough is what looks very straightforward is actually a very twisted process, because DNA is not going to be one straight line. It's going to be very curvaceous. So what's happening is really interesting. You're going in the direction of a 5 to 3 direction, and amazingly, it all goes in that direction. So even though it may seem like, okay, if one's going the opposite direction, they're all going the 5-3 direction. So what happens at the end of replication? The ends of linear chromosomes are called telomeres. They contain repeats of 6 base sequences. So attached to the 5, you're going to have TT, A, GG, G, for example, 6 base sequence. As the replication fork nears the end of the linear chromosome, the lagging strand makes an RNA primer for the end of the chromosome. DNA polymerase 3 uses the available 3-OH hydroxyl group on the RNA primer and polymerizes the last Okazaki fragment for that chromosome. The RNA primer is removed. In the dividing cells, the residual strand DNA is degraded and the telomere is shortened. And this shortened telomere is associated with aging. DNA damage. So DNA replication must be really careful in avoiding making mistakes. And so when there are mistakes, it has to immediately fix it before the replication is complete. Polymerase 3 is found to make the most common mistakes in E. coli during proofreading. This involves the removal of unwanted nucleotides, as known as exonuclease. If a DNA polymer leaves a mistake, enzymes like MUT-S identifies the unmatched base and fixes it. Overall, polymerase 3 mistakes are about 1 out of 100,000. It's pretty good. Repairing damaged DNA. So what are causes of mutations and changes? Well, you can blame UV, smoke, and toxins. UV light damage is called thymine dimers, or I cannot say this, but I'll try. Silobutane pyrimidine dimers. And nucleotide excision repair involves identifying this mismatched dimer, cutting off the mutant, and allowing DNA polymers and DNA ligase to fill in the region. Other types of mutations, we have something called XP, and it's an inherited disease in humans and results to ultra-sensitivity to UV light. It destroys excision repair and enzymes. Then you also have something called TTD and Cockayne syndrome, which is going to weaken the DNA repair mechanisms, resulting in brittle hair and nails. Moving on to vocab review. So what is in a DNA backbone? DNA backbone is going to consist of ribose, phosphate, and bases. What's complementary base pairing? Complementary base pairing is when specific nitrogenous bases bond with each other. Very straightforward. Again, the most common hypothesis today, or to date, is called the semi-conservative replication, where each new chromosome is composed of one strand of old DNA and one newly synthesized DNA. Conservative replication is completely new chromosome. Dispersive replication is going to be new and old sections of DNA mixed randomly. Polymerase 1 is going to remove the DNA primer and replace it with DNA. Polymerase 3 is going to extend an Ozaki fragment, and it's going to perform the bulk of the work. A replication fork is the structure that moves forward from the site of origin. It's that Y shape, basically where everything happens. And then DNA is composed of 3 key components. Again, one ribose, phosphate group, and a nitrous base. I accidentally put that twice. Helicase. Helicase is a catalyst. It's going to break hydrogen bonds between base pairs and open up the double helix. Then we have the SSBP, single-stranded DNA binding proteins that are going to come and stabilize the single-stranded DNA. We have telomerase, which is going to break and rejoin the DNA double helix to relieve twisting forces. You have the primase, is the catalyst and synthesis of RNA primer on an Ozaki fragment. You're going to have DNA polymerase 3, which is going to extend the Ozaki fragment. You have a sliding clamp that holds DNA polymers in place during strand extension, which I didn't really talk about too much, just it's a clamp, holds things in place. DNA polymerase 1 removes RNA primer and replaces it with DNA. DNA ligase is going to be a catalyst joining Ozaki fragments into a continuous strand. Nucleotide excision repair is an example of DNA repair. XP is an inherited disease in humans. It involves sensitivity to light. Excision repair is basically caused mutation of one or several enzymes being mutated. And I already explained what XP is, so I'm not going to go into too much detail on that one. And I also explained what TDD and Cockayne syndrome was. Temperature-sensitive phenotype. Mutant enzyme works at normal temperatures, but at slightly elevated temperatures, it's unable to function because of its mutation. That's pretty terrible stuff. But that concludes DNA replication.